So this chapter is organic chemistry. Organic chemistry revolves around the study of carbon. As an organism, you are what you eat. If you look at any nutritional fact on the back of any of the food you're eating, you'll see all the organic compounds or macromolecules that we'll be studying in this chapter. Fat would be lipids. You see your carbohydrates, proteins, and then nucleic acids would be in anything that was once living, because that's your DNA. So looking at carbon, why is carbon so important? Carbon is the element found in all organic compounds. All carbs, all lipids, all proteins, all nucleic acids have carbon in them. It's also found in carbon dioxide, which is an inorganic compound that is also very important. We'll be studying more about carbon dioxide when we get to our photosynthesis chapter. Carbon forms 4 covalent bonds with other elements. This is what makes carbon so magical. So to the left here, you see the Bohr model for a carbon atom. You see 6 protons, there are 6 neutrons, and there's also 6 electrons. 6 electrons, those are going to be found in the outer shells. So in the innermost shell, you have 2 electrons that can fit. And in the outermost shell, you could fit up to 8. So since carbon has 6 total electrons, there's 1, 2 in the first shell and 1, 2, 3, 4 in the second shell. Now, we could say that carbon has 4 valence electrons. Atoms want 8 electrons in that outermost layer. So we need 4 more to make carbon a happy atom. So carbon's going to be able to covalently bond using each of these 4 electrons to make 4 bonds with other elements. So this allows carbon to bond to itself to form chains. These chains could basically be an infinite number in length. So you get carbon, bond to carbon, bond to carbon over and over and over again. These chains can then branch off from each other and make more chains, and then the chains can loop in on themselves to form rings. These rings could be branched out into chains, into more rings. You could have all these different combinations. So it explains the large variety and number of organic compounds that we have. So there are 4 different macromolecules that we're going to look at in this chapter. The first one is carbohydrates. The second is lipids. Third is proteins. And 4 is nucleic acids. Nucleic acids we will have an entire chapter on coming up later on in the semester. So we're not going to go in too much detail on that, but the other 3 we will get into detail on. The video at the bottom is an Amoeba Sisters video. If you want to check that out, I could post that link for you. I love the Amoeba Sisters. So first up, we've got our carbohydrates. Carbs are all of these wonderful things that you see on the screen. You've got your fruits, and you got some vegetables there, your breads, your pastas, your potatoes, your rice, things like that. Carbohydrates can be abbreviated as CHO. That's because they are composed of carbon, hydrogen, and oxygen. If you were to look at the chemical structure of a carbohydrate, the proportion of hydrogen to oxygen is always 2 to 1. So for every 2 hydrogens, there's going to be 1 oxygen. The reason we care about the ratio of hydrogen to oxygen is because when we get to lipids, lipids are also made up of carbon, hydrogen, and oxygen. So looking at the ratio of hydrogen to oxygen is how we're going to be able to tell the difference between carbs and lipids. Carbs provide you with energy. That is the main thing that they do. So people who try to cut out carbs from their diet, they often feel very tired because they're not getting that immediate energy that people who eat carbs are getting. Carbohydrates are going to be subdivided into 3 groups that we're going to be talking about. The first one, monosaccharides. Remember, mono means 1. So these are 1 very small carbohydrate. Disaccharides. Di, the prefix di means 2. So disaccharides are 2 monosaccharides bonded together. And polysaccharides. Poly means many. So this would be many monosaccharides bonded together to form 1 very large complex carbs. Your mono and your disaccharides are your sugars. Sugars are carbs. So anything that you're eating with sugar is technically considered to be a carb. So even if you're on a diet and you decide that for lunch you're going to eat an apple because you want to cut down on your carbs, there are still carbs in that apple. Your polysaccharides are your complex carbs, like your starch. We'll talk about glycogen and cellulose as well. So let's get into this. Here's a picture showing you the difference between mono, di, and poly. And we're going to start with monosaccharides. Once again, these are your simple sugars. Their monomers can have between 3 and 9 carbon atoms, but the ones we're going to look at, the most common, have between 5 or 6 carbon atoms. Monosaccharides are going to provide you with quick energy because they're very small. Because they're small, they're easy for your body to break down. But that energy that you're getting from the monosaccharides is only going to last for a short period of time. This is why a lot of people, when they eat these monosaccharides only, they're experiencing a crash after their body burns these monosaccharides, because then that energy has been depleted. These are things like your fruits, your vegetables, Gatorade, Powerade, fruit drinks, energy drinks, candy. Once again, that quick but very short-lasting energy. Common monosaccharides that we'll be talking about: glucose, fructose, and galactose. Notice they are all C6H12O6. In class, we talked about how these are all isomers. They all have the same ingredients, same molecular formula, but their structures are different. So I used the Lego example in class. If I gave 2 students each 3 red Legos, 3 yellow Legos, and 3 blue Legos, and I asked them to build a tower without looking at each other's products, they probably wouldn't be building the same tower. Same thing here. They have all the same ingredients, the same Legos, but they're put together differently and are therefore different sugars. Here are pictures of their structures, so you can see that they do look different even though they are made of the same things. 2 other monosaccharides that we'll look at in this chapter and other chapters that will be coming back to us would be deoxyribose sugar, which is found in DNA. It has 5 carbons. And ribose sugar, which is found in RNA, which also has 5 carbons. We'll study those more when we get to our nucleic acid chapter. Disaccharides are next. Di means 2. So once again, these are 2 monosaccharides bonded together to make a disaccharide. The process that is going to build a disaccharide is called dehydration synthesis. So we add 2 monosaccharides together. We want to bond them together. In order to do that, we need to remove water from their chemical structure. When we remove water from their chemical structure, we get our disaccharide. And then we obviously have to show water as our product. Once again, that is dehydration synthesis. So here are a couple examples. If you add 2 glucose molecules together and remove the water from their chemical structure, they will bond together to form maltose, which is malt sugar. So anyone that has a malt shake instead of a milkshake, it's made with a different type of sugar. It feels grittier. If you add a glucose and a fructose together, you're going to get sucrose, which is a table sugar. This is the sugar that you'd probably have in your house, in your sugar bowl, or in your canister. So we want to add these 2 things together. You remove the water from the structure. We show it here as a product on the right side of the equation. And we've made a bond to make sucrose. And our last one here, glucose and a galactose. When we put those together, dehydration synthesis, we pull the water out, show it as a product. And that will make lactose, which is milk sugar. Some people are lactose intolerant, which means they cannot break down lactose. They don't possess the chemicals in their body to break that down, and it makes them very sick. Showing it to you in an actual chemical equation format with molecular formulas, you see 2 monosaccharides. They are both C6H12O6. We want to basically add those 2 things together, bond them together to make a disaccharide. So when we put those together, we get a disaccharide, which is C12H22O11, and water. So if you look at adding these things up, 6 carbons and 6 carbons, 6 and 6 is 12. 12 hydrogens and 12 hydrogens, 12 and 12 is 24. But here you see 22. Where are those other 2 hydrogens? Over here in the water. Because remember, we had to pull water out of the chemical structures on the left. 6 oxygens and 6 oxygens, 6 and 6 is 12. But over here you see 11. Here's that 12th one there. So you actually pulled that water out of these 2 monosaccharides. If we wanted to break down those sugars, we'll see hydrolysis in a second. But here are the structures of these 3 sugars. Maltose, sucrose, and lactose are also all isomers, which means that they have the same molecular formula, C12H22O11, but they are different sugars because they are all put together differently. You'll also notice that all of our sugars, maltose, sucrose, lactose, glucose, galactose, fructose, deoxyribose, and ribose, they all end in OSE. Science does that, where we like to try to group things together, so they all end in ose. If we ingest a disaccharide and you want to break, your body needs to break that down, that process would be known as hydrolysis, hydro water, lysis break apart. So we're going to take our disaccharide, add water to it. In that process, we break the bond really between these two monosaccharides, and we get our two small pieces back. So here is what it looks like for each of our disaccharide sugars. You take maltose, you add water to its structure. It'll break the bond here and break maltose back down into its two glucose molecules. That's the same for sucrose and for lactose. So once again, that process is known as hydrolysis, hydro water, lysis to split. You're breaking down by adding water to the structure. Here are some videos that I showed on dehydration synthesis and hydrolysis. So I'll just click on that here to show you. So this is loading, taking some time here. But here you could see that you have two, I want to start that over here. You have two monosaccharides. We want to put them together. So when they interact, you're going to pull the water out, form a new bond, and that will make our disaccharide. Disaccharides are larger and more complex than monosaccharides and therefore contain more energy. They do take longer to break down or digest than monosaccharides, but they're still very small, and your body will burn through them pretty quickly. However, it does last longer than your monosaccharides. That's it for disaccharides, and we're going to move on now to polysaccharides. Polysaccharides are composed of 3 or more monosaccharides bonded together by dehydration synthesis, and once again broken apart by hydrolysis. So 3 or more. So we have 1, 2, 3 monosaccharides. We want to build bonds between those monosaccharides. So we need to take the water out. Okay. When we take the water out of these structures, we're able to make these bonds. We show water as a product. If we wanted to break down a polysaccharide, we add the water into this structure to get our individual pieces back. 3 common polysaccharides. You got your starches, you got your glycogen, and you got your glucose. Starches are polysaccharides produced by plants like potatoes, corn, rice, wheat, and other grains. They are just really, really long chains of glucose molecules, and they could be very, very long in length. So you'll see here the 3 dots at the end. That signifies that the chain can go on and be longer. Glycogen is a polysaccharide produced by animals. It's made of many glucose molecules bonded together in branched chains. So it's not just one chain, it's many chains of glucose. This is produced in animals' liver and stored in your liver and in your muscles. So you are able to make this and store this. So once you burn through your mono and your disaccharides, you would then pull from your glycogen. That would be your second wind of energy. It provides long-term or long-lasting energy. So this is going to give you more energy than your mono and your disaccharides for sure. Cellulose is produced by plants. It consists of chains of glucose that are bonded horizontally and vertically, which makes this net-like structure. Because of this structure, cellulose is very strong. It gives plants strength and support. And because it's so strong and that net-like structure is present, humans and other animals, we cannot use cellulose for energy in its raw form. You need to cook celery or cook broccoli in order to get energy out of it. In its raw form, we cannot break those bonds down completely. We don't have the enzymes or the chemicals needed to do this. So it's a source of bulk or fiber for us, which means it's going to run through our digestive systems, kind of clear out our digestive systems and help you go to the bathroom. Number 2. That brings us to lipids. So we're going to stop the video here and go on to the next one.